This project investigated the effects of antimicrobial peptides on Pseudomonas aeruginosa and Staphylococcus aureus contact lens biofilms. Biofilms are communities of microorganisms that grow on a substrate. Bacteria in a biofilm show increased resistance to antimicrobials compared to bacteria in their planktonic state. Contact lenses can introduce foreign microbes onto the ocular surface and act as a substrate for biofilm growth, which can lead to bacterial infections. Pseudomonas aeruginosa and Staphylococcus aureus are two prevalent microbes that cause ocular infection. The eye has a variety of defense mechanisms to protect against infection, such as the physical barrier of the eyelid and eyelashes, the presence of ocular mucins that prevent bacterial attachment, and the enzyme lysozyme in the tear film, which breaks down bacterial cell walls. Another ocular defense mechanism is antimicrobial peptides, which are released as an innate immune system response. AMPs are sequences of L-amino acids that can take on a variety of secondary structures. The most prevalent AMP structures on the ocular surface have beta sheets and alpha helices. AMPs inhibit bacterial growth by disrupting cell wall formation, interrupting cell signaling, or downregulating genes that promote biofilm formation. The AMPs used in this study are HNP2, which is shown in the figure on the left, and HBD1, which is shown in the figure on the right. One of the most prevalent AMPs on the ocular surface is cathelicidin LL37. It is expressed in the glands of Moehl, which are the modified sweat glands, the cornea, which is the refractive covering over the pupil and iris, and the conjunctiva, which lines the eyelids. Defensins are the other major AMP on the ocular surface. There are two categories of defensins: human neutrophil peptides, abbreviated HNP, and human beta defensins, abbreviated HBD. Human neutrophil peptides are expressed in the tear film, and human beta defensins are expressed in the tear film as well as ocular tissues, including the glands of Moehl, the cornea, the iris, the conjunctiva, the lens, which focuses light entering the eye, ciliary bodies, which support the lens, and both the vitreous and aqueous humor, which are the fluids within the eye. Current treatment for ocular infections relies on antibiotic drops. However, with antibiotic-resistant bacteria on the rise, it is essential to have alternative treatment methods in place to treat antibiotic-resistant bacterial infections. This study investigates human neutrophil peptide 2 and human beta defensin 1 as potential treatment options. Because human beta defensin 1 is expressed by more ocular tissues, it is hypothesized that it will be more effective at inhibiting bacterial growth and biofilms. Because of the limited amount of AMPs, experimental wells were scaled down to 50 microliter volumes. The purpose of the initial bacterial strain testing was to ensure uniform growth at the 50 microliters compared to 100 microliters. 2 strains of P. aeruginosa were tested, 1 strain of S. aureus was tested, and 1 strain of E. coli was tested. The bacteria were grown in a 96 well plate in a 1-to-1 ratio with TSB at either 50 or 100 microliters. And following incubation, absorbency was read at OD 600. The image shows the plate set up for these trials. Note that at the 100 microliter volume for P. aeruginosa, the color of the wells are green. This is because P. aeruginosa has the ability to quorum sense. Because the AMPs get resuspended in 10 millimolar acetic acid, I ran the acetic acid MICs to determine if acetic acid had an effect on bacterial growth. Bacteria was grown in a 1-to-1 ratio of serially diluted acetic acid and TSB, and after incubation, absorbency was read at OD 600. To begin AMP testing, Kirby-Bauer disc diffusion susceptibility tests were run. In that test, 6 mm filter paper discs were treated with stock AMPs and then transferred onto a plate with bacteria. Following incubation, if the bacteria is susceptible to the AMP, a zone of inhibition, which is demonstrated by clearing around the disc, will be seen. Then an AMP MIC was run to determine what concentrations of the AMPs affected bacterial growth. Like the acetic acid MIC setup, bacteria was grown in a 1-to-1 ratio of serially diluted AMP and TSB. Following incubation, absorbency was read at OD 600. Then biofilm staining using 0.1% crystal violet was done, and then absorbance was read at OD 450. The last procedure were for the biofilm disruption trials, in which pre-formed P. aeruginosa biofilms grown on contact lenses in 6 well plates were treated with AMP, acetic acid, or saline for the growth control. Samples of the planktonic solution were taken every 20 minutes after the initial treatment. At the end of the 2-hour treatment period, the lenses were swabbed. The microdilution plating technique, in which serially diluted samples are plated on dry TSA plates, was used to quantify results. For the preliminary testing, there was no difference in absorbency between the 50 and 100 microliter volumes for all strains except P. aeruginosa BAA26, which had significantly lower growth at the 50 microliter volume compared to the 100 microliter volume. However, even though the absorbency was lower, its growth was uniform at the 50 microliters, and so it was selected as the gram-negative bacterium moving forward. S. aureus was confirmed as the gram-positive bacterium. For the acetic acid MIC, none of the tested concentrations completely inhibited bacterial growth. There were no clear wells. But there was a difference in absorbance readings at the 2.5 millimolar concentration for P. aeruginosa. For the Kirby-Bauer, all bacteria were resistant to both amps. As seen in Figure A, there are no zones of inhibition for P. aeruginosa. No zones of inhibition were reported for S. aureus or the mixed culture either. However, there was a reduction of growth around the discs on the P. aeruginosa plate, as shown in Figure B. So I went ahead with the AMP MICs. After one MIC for each AMP, only human neutrophil peptide 2 was selected for further testing due to the financial constraints of the project, even though human beta defensin 1 resulted in lower absorbencies. This graph compiles the acetic acid MIC data and the human neutrophil peptide 2 MIC data. The average acetic acid absorbance value for each concentration was subtracted from its corresponding average AMP absorbance value. The difference in absorbency is on the y-axis, while the AMP concentration is on the x-axis. Blue bars represent S. aureus, orange bars represent P. aeruginosa, and gray bars represent the mixed culture. The positive values mean that the difference is due to the AMP, but a negative value means that the difference is due to the acetic acid. So at the lower concentrations of the treatment, the AMP is the reason for the difference in the absorbency readings, but at the higher concentrations of treatment, the reason is the acetic acid component of the treatment. These graphs show that the colony count results from the end of the 2-hour treatment interval for the biofilm disruption trials. Purple bars represent the AMP treatment, gray bars represent the acetic acid control, and yellow bars represent the growth control. Part A on the left shows the untransformed data, but because it was not normally distributed, Part B shows the log transformation, in which there is no significant difference between the AMP treatment and the controls. However, please note that the AMP treatment average reflects only 3 contact lenses. Furthermore, the acetic acid and growth controls reflect only 1 lens each, which is why no standard deviations are reported for them. There is also no significant difference in colony counts between the samples taken in 20-minute intervals. P. aeruginosa is capable of quorum sensing. When a cell density threshold is reached, specific genes are activated that benefit growing in high populations. During the strain testing, P. aeruginosa was able to quorum sense at the 100 microliter volumes, but not at the 50 microliter volumes. That explains why growth was significantly lower at the 50 microliter volumes compared to the 100 microliter volumes, and this was confirmed by the green color of the 100 microliter wells. Furthermore, P. aeruginosa outcompeted S. aureus in the mixed culture, as the color of the mixed culture was green like pure P. aeruginosa. Throughout the MIC testing, the mixed culture reported similar absorbency values to P. aeruginosa. Acetic acid is known to inhibit bacterial growth by disrupting cell walls. Previous studies found an acetic acid MIC value ranging from 0.16 to 0.31% for S. aureus and P. aeruginosa isolates. An acetic acid MIC of 0.31% corresponds to a molarity of approximately 3 millimolar. The highest acetic acid concentration used in this study, 2.5 millimolar, is lower than the highest published MIC results, which could explain why no acetic acid MIC was found. However, the results of this study do support previous research that acetic acid reduces bacterial growth, especially at high concentrations. No MIC was found for either AMP, contrary to previous studies which found defensin MICs ranging from 5 to 10 micrograms per milliliter. However, to prevent evaporation during incubation, the 96 well plates were wrapped with parafilm, which potentially created a microaerobic or anaerobic environment. Though S. aureus and P. aeruginosa are facultative anaerobes, meaning that they can grow in both aerobic and anaerobic conditions, they should have not been affected in a low oxygen environment, but the AMP's potency could have been altered. A previous study had shown that the ability of AMPs to inhibit bacterial growth decreased in anaerobic environments. For the graph depicting the effects of acetic acid compared to the AMP, no statistics, including standard deviation, could be reported since the acetic acid MICs and the AMP MICs were run on different days. More accurate results would be shown if they had been run on the same day from the same overnight culture, and statistics could then be applied. However, as presented, the data suggests that had acetic acid not been the solvent for the AMPs, not as much bacterial growth inhibition would have been seen, since the reason for the decrease in growth for the highest concentrations of AMPs was because of the acetic acid component of the treatment. While this study showed no difference in growth for the 2 hours following initial treatment, other studies did note a decrease in growth for S. aureus around 60 minutes after treatment and 100 minutes for P. aeruginosa against human neutrophil peptide 1. Perhaps the treatment period for this study should have been extended to over 2 hours in order for a trend to emerge. In conclusion, this study suggests that AMPs cannot be used to effectively eradicate contact lens biofilms or consequently be used as treatment for ocular infections. However, to get more accurate data, increasing the number of replicates of experimental and control groups per trial, as well as increasing the number of trials overall for both the MIC and contact lens components of the study, should be done. Any further experimentation on AMPs should consider using synthetic AMPs rather than naturally derived ones. Partnering with the biochemistry project would allow for a larger quantity of AMPs to be available at a less expensive cost, and higher concentrations of AMPs could be tested. Lastly, because AMPs on the ocular surface interact with the tear film, perhaps testing the AMPs in combination with other components of the tear fluid would more closely mimic physiological conditions and yield more promising results. The A.J. Palumbo Student Research Endowment Fund provided the funding for this project. The St. Vincent Biology Department provided further funding, as well as the equipment and other resources needed to carry out this experiment. I would like to thank my advisor, Dr. Jennifer Koehl, for her guidance, support, and dedication throughout the project. And I'd also like to thank the Koehl staff research students, especially Kaitlin Schiffer and Michael Sella, for their assistance throughout this project. Here are the references used throughout this presentation.